The anatomy of cardiac muscles is skeletal muscles, and the cardiac muscles, they are also elongated, but they are branched. Skeletal muscle are non-branched, and they contain these cardiac muscle. They contain usually one and sometimes two nuclei that are located in center. Cardiac muscle cells contain actin and myosin myofilaments like in skeletal muscle, and that are organized in same type of sarcomeres. The actins, the thin protein, they are overlapping thick protein myosin, and the arrangement is similar to that of skeletal muscles. And the because arrangement is similar to skeletal muscle, so the cardiac muscle, they also appear banded or striated in under microscope, but the striations, they are less, and they are less regular as compared to skeletal muscle. Cardiac muscles, they have smooth endoplasmic reticulum, but it is not having a surface very well developed along T tubules, and the cardiac muscles cells, they have connection between them. These connections, they are we can see here, they are folds in membrane, and they are called as intercalated discs. The junction between two cells of cardiac muscle or cardiac muscle cells, there are junctions. These junctions, they are called as intercalated discs. These are the folds of membrane, and then inside these folds there are uh, two types of cellular junctions. The gap junction, we know the gap junctions are the protein bridges between cells. So cells, they exchange material through these gap junctions. And there are another type of connection that is called as desmosomes. So the desmosomes, we know they are glue like connection that are these are also made up of glue like protein that attaches to membrane together. So the intercalated discs, they are, we can say, made up of three type of connection. One connection is physical through the folds of membrane, so that the two membranes of two cells, they fit together through these folds. And then there are gap junctions. Gap junctions are bridges through which material can pass from one cell to another cell. And then there are desmosomes, that are the glue like protein that attaches two cells together. Because there are gap junction, cells can pass material from one cell to another cell. So the cardiac muscle, they behave as a single unit. They are highly coordinated, and they produce coordinated contraction of the heart, and that is required for the functions of heart in order to pump the blood in synergic way. So if action potential is produced in one cell, it can be relayed on to other cell through these gap junctions. Similar to skeletal muscles, the cardiac muscles, they have uh, T tubules that are the invaginations of cell membrane or sarcolemma, and these T tubules, they come in contact with sarcoplasmic reticulum inside the cell, and there are formation of triad. We know this triad is present in skeletal muscle, that there are two endoplasmic reticulum or sarcoplasmic reticulum are present on both sides of a T tubule. So this whole structure is made up of three components: two endoplasmic reticulum and one T tubule is called as a triad, made up of three things. So this T tubule is part of cell membrane. So when action potential runs on membrane, it goes to the T tubule, and then it influence these endoplasmic reticulum to release their calcium. But in case of this cardiac muscle, T tubule is coming into the cell on the Z disc of the sarcomere. Z disc of the sarcomere, where the sarcomere, one sarcomere ends. Here we can see uh, here is a Z disc and another Z disc. We can see two blue color uh, transverse tubules, they are coming into the cell at Z disc, and here we can see actin and myosin, they are overlapping on each other. This central line is M line, that is the middle line or the middle line. So the in case of skeletal muscle it is different, because the T tubules they come on the center of the sarcomere, that is where the actin and myosin joins. So calcium reaches directly to the place where it has to attach with troponin. But in case of cardiac muscle, the calcium has to travel distance to go to the actin and myosin, so it takes time for the contraction. So the cardiac muscle, they are slow contractile. And secondly, because the endoplasmic reticulum or sarcoplasmic reticulum, they are not that well developed as that in skeletal muscle. So calcium, few of the or we can say a significant amount of calcium has to come from extracellular material. So this calcium, entry of this calcium takes time, so there will be delay in contraction of the cardiac muscle. So cardiac muscle, they are uh, slowly contracting and slowly relaxing because of these factors. Like in other muscles, adenosine triphosphate or ATPs are responsible for the energy of cardiac muscle, and ATP production is dependent on oxygen availability. As we know, if oxygen is available sufficiently, then the metabolism will go to the aerobic respiration, and if the oxygen is not present, then it will remain up to glycolysis. And we know in glycolysis there is production of only two ATPs for use of the tissues. So the cardiac muscle, they cannot hold a lot of oxygen, that so they have a lot of oxygen supply, and that oxygen supply is sufficient to, because they have a lot of blood supply, so oxygen supply is sufficient to produce the energy. And on the other hand, they also have a lot of mitochondria as compared to skeletal muscles, so that they can efficiently use this oxygen and glucose to produce sufficient amount of ATPs in aerobic respiration. So these ATPs, they are always available to the cardiac muscle, because body cannot risk the fatigue of cardiac muscles. So the uh, heart is made up of two types of tissues: connective tissue, that is making the core of the heart, or the base of the heart, or foundation of heart, or skeleton of heart, and the conducting and the contractile system is made up of cardiac muscle that are wrapped onto the connective tissue. So this connective tissue is fibrous connective tissue. It provides the barrier between the right and left part of the heart, that is called as septum or the wall, and it also provides the uh, uh, the plates between atria and ventricles, and it also provide a base for the atrioventricular valves that are between atrium and ventricle, and we know they are bicuspid and tricuspid wall. And bicusp mean they have two lids, and tricusp they have three lids. So then the connective tissue, it also provide the base for the semilunar valves. Semilunar valves are between they are pulmonary semilunar valves, and then they are aortic semilunar valve. So these aortic semilunar valve and pulmonary semilunar valve, they are made up of connective tissue, fibrous connective tissue. The fibrous connective tissue also provide the electrical insulation between the atria and the ventricles, and it provides, as we know, the site for the attachment of the cardiac muscle on it. So the fibrous connective tissue, it is working as skeleton of the heart. We'll see that the conduction system, when it passes from the AV node to the apex of the heart, most of it runs through the septum that is made up of the connective tissue. So it does not come in contact with the cardiac muscle directly when it is coming from AV node. So the contraction starts from apex, where the AV bundle, it enters into the cardiac muscle from the septum. So it is providing insulation or insulation way for the passage of AV bundle. We'll see it later. The cardiac muscles or the heart has a conducting system, and that conducting system is responsible for the spread of action potential on the all the muscles of the heart. And this conduction system is different than other cardiac muscle because it is modified in the form of fibers that are like neurons. So these fibers, they are making the two nodes and a conducting bundle. We know the well known sinoatrial node or SA node, and an AV node, atrioventricular node, and then there are fibers that are atrioventricular bundles, and then these fibers, they give rise to Purkinje fibers at the apex of the heart. So these are the conducting system. They are making two nodes, and then fibers, they are making conducting system of the heart. They are modified type of non-contractile type of the cardiac muscles. So they are uh, for only conduction of the action potential. They are muscle cell, but they are modified in the form of neurons. And the nodes are contained in the wall of right atrium and named according to their position. We know the sinoatrial node, it is medial to the opening of the superior vena cava. So it is in the middle of the opening of superior vena cava. And the atrioventricular node, it is present at the medial to the right ventricular valve. So it is called as atrioventricular because it is in middle in between these two uh, chambers, atrium and ventricle. So these are present in two nodes are present in right atrium. The only AV node give rise to the conducting bundle of the heart, the atrioventricular bundle of the heart. They are also called as bundle of His. This bundle passes through small opening in the fibrous skeleton to reach the intraventricular septum. Here, the intraventricular septum is present. Septum means a wall, the wall that is separating atria, left atria and right atria, and the ventricle, right ventricle and left ventricle. So this wall of the heart, because inside we know is the connective tissue, so this wall is made up of connective tissue, and on that muscles are uh, present. So these uh, atrioventricular node, it is giving rise to atrioventricular bundle, and these atrioventricular bundles, they are bundle of his, they are running in the septum of the heart, or the wall of the heart, that is present between two parts of the left and right part of the heart. Inside the septum, these bundles, they divide into right and left bundle branches, and then they give rise to smaller branches. When they reach to the heart apex, give rise to small branches. These branches then enter into uh, the wall of the muscles of uh, heart, and these branches, they are called as Purkinje fibers. As we know, the atrioventricular ventricular bundle and the right and left bundle branches, they are less contractile, but Purkinje fibers, they are more modified, they are least contractile, and they have a lot of gap junction between them, their cells, so the action potential rapidly travel in the Purkinje fibers as compared to other fibers and the muscles of the heart. All the cardiac muscles, they can generate action potential by themselves. It means they are independent of any input from the nervous system. They can develop their action potential themselves, but the SA node can generate action potential more efficiently than the and a lot of action potential than the other um, cells of the cardiac tissue. Therefore, SA node in a normal heart takes charge of the whole production of whole action potential or stimulus of the other cells of the cardiac muscle. Therefore, it is called as a pacemaker of the heart, and it is responsible for the spontaneous and rhythmic contraction of the all the heart uh, muscles. When there is production of action potential on sinoatrial node, then it is separated to AV node, and they say there is a preferential pathway, but it is not discovered yet. So that preferential pathway transfer action potential from atria, from SA node to the AV node. And when heart is beating at a normal rate, the action potential takes about 0.04 seconds to travel from SA node to AV node. In AV node, the action potential is uh, propagated a little delayed, and there is delay of about 0.11 second from the time action potential reaches the AV node until they pass to the AV bundle. So that time is 0.11 second the AV node takes to deliver this action potential to atrioventricular bundle. Therefore, 0.04 second here and 0.11 second from AV node to the AV bundle. This is a total 0.15 second time that is required atria to contract before ventricle. So we can say atria contract 0.15 second before the contraction of ventricles. When AV node sends action potential to atrioventricular bundle, then it goes to left and right bundle, and then it reaches to the Purkinje fiber. When it reaches to Purkinje fiber, it becomes very fast, and then it enters into the cardiac muscle cells. And when it is relayed on the cardiac muscle cell, this area of the heart is pointed area is called as apex, and this broader area is called as a base. So in apex there is because Purkinje fibers are directly entering into the uh, heart tissue or myocardium. So the action potential is now relayed on the cardiac muscle cell. So they start to contract. So heart start contraction, or the ventricle, they start contraction from the apex to base. So the heart become smaller from apex to base because it contracts upward toward base. So we can say atria, they contract from base to apex, when action potential is produced from SA node, and it is relayed on atria and it is relayed on AV node. And after 0.15 second, because the action potential takes to reach to the AV node, and then AV node relay it on the atrioventricular bundle. Up to that, there is a delay of 0.15 second. After that, the action potential enters into the Purkinje fibers, and from Purkinje fibers the muscles, they get action potential, and then they start to contract. And contraction of ventricle is from the apex to base. The resting membrane potential in muscle cell is similar to other cells, that the inside of the cell is more negative because it has more negative ions as compared to outside of the cell. We know inside there are more DNA, RNA and protein that have negative charges, and they are not present in extracellular material. And then the membrane is less permeable to sodium and potassium and calcium, because calcium and sodium ions, they don't have open channels in cardiac muscle and other cells. So sodium and calcium, they are more concentrated outside, they cannot come into the inside the cell to make it more positive from inside, so cell remain negative from inside. And then there is more permeability to potassium, because there are certain potassium open channel present, and we know open channel, they can conduct potassium anytime. So potassium keeps on leaking, and inside becomes, because normally potassium is more concentrated inside, so it is leaking through the open channels along its concentration gradient, so making inside more negative. The action potential produced in cardiac muscle cell depends on type of cells. For example, there are two different types of cell, like all muscle cells, and then there are nodes. Because nodes are autorhythmic, so their action potential is different than the other muscle cell. We have to describe both processes in detail in the next slide. The heart is autorhythmic muscle, so it can contract on its own. It can produce its action potential itself and contract its uh, muscles. And if we remove heart from the body and maintain all the physiological conditions, that are the temperature, nutrients, gases, and the ion concentration, then it can maintain itself and can work like it is working inside the animal body. So it is called as autorhythmic because of presence of SA node and AV node. The SA node produces the action potential on itself, and it relays it onto the atrial wall, and then to AV node, and then onto the AV bundle. And SA node is the main in charge of the action potential. It causes the other cells to open their sodium channels, uh, and it sends action potential through the intercalated disc between the cells, so action potential spreads on the whole membrane of the whole cells, all the cells of the whole heart. The production of action potential in the SA node is very interesting. and it is spontaneous, it is produced on itself. We know that the resting membrane potential, the inside of membrane is negative, and the concentration of sodium is more outside, calcium is present more outside of the cell membrane, and potassium is present more inside of the cell. So when the membrane is on resting membrane potential, in case of the SA node, there are present sodium open channels. So sodium open channels, they are not present in other cells, they are only present in the SA node. So these channels start to conduct sodium into the cell. So sodium start to come into the cell, making it positive from inside. So this change in potential, that is inside, goes a little positive due to entry of sodium from sodium non-gated or open channel that are not present in other neurons and other cardiac muscle cells or skeletal muscles on which action potential usually runs. It is only present on the SA node and sometimes AV node. So on the SA node, generally we are talking about SA node, we'll talk about AV node later. As in SA node, the at resting membrane potential, sodium open channels, they start to conduct sodium into the cell because sodium is more concentrated outside, so there is a gradient to to conduct sodium into the cell. So sodium comes into the cell, makes it a little positive, that is called as a pacemaker potential, or it is called as a pre-potential. It is called as pre-potential or pacemaker potential. So this pacemaker potential opens two types of channels. It opens calcium channel here and the sodium voltage channel, calcium voltage channel and sodium voltage channel, this pacemaker potential or pre-potential. And calcium channels get open and sodium voltage channel, they get open, and sodium and calcium, they enters into the cell, makes membrane more positive, that is called as depolarization. So depolarization only in pacemaker cells of the heart are the responsible of depolarization. Calcium in other cells like skeletal muscle, in neurons, and the other cardiac muscle that are contractile cardiac muscle, calcium is not responsible for depolarization. We saw calcium is responsible for a plateau phase, but sodium is causing depolarization. In this case, in pacemakers SA node and AV node, the sodium and calcium, they simultaneously cause depolarization, and then the repolarization is caused by opening of potassium channel. So potassium leaks out and calcium channels, they get closed, and sodium channel, they get closed, so there is a repolarization. And then we know potassium channels, they are slower channel, so they will bring membrane back to the resting membrane potential and a little more negative. And then again at negative potential, that is about minus 60 millivolt, these pacemaker cells, they, because they have sodium open channels, so sodium start to come into the cell. When inside become negative, this attracts sodium to enter into the cell. So sodium enters into the cell, there is production of a little positive charges. These positive charges are called as pacemaker potential or pre-potential, that is then responsible for opening of calcium voltage channel, sodium voltage channel, that produces a depolarization again. And then repolarization is done by the opening of potassium voltage channel, that opens at positive charges, and then they conduct potassium outside of the cell, and again it goes to repolarization, and there is closure of calcium channels. So action potential in pacemaker cell, that SA node and AV node, is different than the other cardiac muscle cell, because depolarization is done by calcium and sodium, and the pre-potential is produced by sodium open channel. So these sodium open channels are responsible for they are also called as funny channels, because they are producing action potential when there is no stimulation from outside. So they keep on producing action potential. If SA node does not work on a heart, then AV node takes its charge. It has same type of autorhythmicity, so it can maintain rhythm, but in a little lesser efficient way. And if AV node also cannot work, then this spread on the whole membrane, whole heart muscles start to contract in their own rhythm. They cannot maintain rhythm. They start to contract in their and produce action potential by themselves. So there is a um, condition that is called as arrhythmia, mean rhythm is not maintained. So arrhythmia cannot contract the heart in a synergic way, in simultaneous way, so it cannot produce pumping force. So the function of heart is compromised. In that case, the pacemakers with a battery are installed into heart to produce a rhythmic action potential in order to get an arrhythmic contraction of atria and ventricles. The production of action potential in cardiac muscle, the contractile cells of the cardiac muscle is similar to that of neuron and skeletal muscle. We know the resting membrane potential in cardiac muscle is at minus 90 millivolt, and it is due to the presence of protein and DNA, RNA, and presence of more positive ions outside as compared to inside. So inside is negative, that is minus 90 millivolts. Sodium and calcium, they are more outside, and potassium is more inside, but potassium tend to leak out because of open channels, so making inside more negative. So when there is depolarization, it is entry of sodium in other cells, but in case of heart, there is involvement of calcium. And the action potential in cardiac muscle, it lasts longer as compared to skeletal muscles and neurons. And the because the skeletal muscle cell, they conduct action potential in 2 milliseconds, and the heart muscle, they conduct action potential in 200 to 500 milliseconds. So action potential in cardiac muscles is slow to complete as compared to the action potential in the skeletal muscles. In the cardiac muscle cells, the contractile cells, the resting membrane potential is minus 90 millivolt. And when we know the in case of skeletal muscle cell, all the cells, they are independent, so they need a neuron and axon coming on to them and making neuromuscular junction on each cell in order to stimulate it or produce action potential in it. We know acetylcholine is released from a skeletal muscle, that is a neurotransmitter, it stimulates the skeletal muscle. But in cardiac muscles, the autorhythmic cells, that are the AV node and SA node, they may conduct action potential in the bundles, and these atrioventricular bundles and Purkinje fibers, they then make connection with the cardiac contractile muscles through gap junction. We know there are the intercalated discs. These intercalated discs, they have three characteristics. They have fold cell membrane in order to fit with the neighboring cell, and they have gap junction between them, that are bridges through which the material can pass from one cell to another. And then they have desmosome glue like protein. So they have gap junction, they can pass their positive ion directly into the contractile cells, and from one contractile cell to another contractile cell, because the contractile cell, they also have in between them the intercalated discs that have gap junction or bridges. So if one cell is uh, having action potential are stimulated, then it can pass its ions directly to the neighboring cell. So if this cell was on minus 90 millivolt, it was on resting membrane potential, and it has got few sodium ions from the neighboring cells because that has gone to action potential. Then these uh, the sodium voltage channel in this cell, they are uh, sensitive to the sodium entered from neighboring cell, and these sodium voltage channels, they start to open, and when they open they are called as fast channel in case of heart. So they open very fast and sodium enters into the this cell and making it more positive from inside. So this cell is called to be depolarized now, because it is going from minus 90 millivolt, that is a larger magnitude, toward plus 20 millivolt. It will pass from 0 and then stay at plus 20 millivolt. Why it stays at plus 20 millivolt? But because at plus 20 millivolt the sodium channels, their voltage channel, their inactivation gate, they get shut, and no more sodium can enter into the cell. This phase is called as rapid depolarization of the contractile muscles of the heart, because of the opening of sodium voltage channel and influx of sodium into the cell. And then there is a little depression, that there is a little repolarization, and it is similar to that of the depolarization is similar to that of other cells like skeletal muscle cells and the neuron. Here we see the membrane is at minus 70 millivolt, and then there is entry of sodium. Here we can say, see, the sodium enters and the cell become positive. It is called as depolarized. It was at minus 70, now it has gone to minus 30, or sorry, plus 20 or plus 30. So it is called as depolarized because magnitude of ions has been decreased, because it was minus 70, now it is only plus 30. So we have to keep it, remember, it is not the plus and minus quantities, these are the plus and minus charges. So plus charges mean the plus the charges, ions that have positive charges, and minus 70 mean the ions that have minus charges. It is not the magnitude, related to the magnitude of quantities. We know in case of quantities minus 70 is smaller than plus 30 in case of quantities, but these are not quantities, these are charges. Plus it is 70 and 30 are the quantities, so 70 is always greater than 30, but it is, these are negative charges, so it is called as minus 70, and the plus 30 are the positive charges, so it is called as plus 30. So now we know that minus 70 is greater than plus 30 because magnitude is 70 and 30, so it is called as depolarization. It is similar to that of the cardiac muscle. And then in case of skeletal muscles and neuron, there is repolarization due to opening of potassium channel. But in case of cardiac muscle, there is a repolarization, a little depression, and that is due to opening of potassium channels. But potassium channels, they are slower channels, and then there are calcium channels also present in cardiac muscle cells. They are slow, they are also slower channel, so they get open after potassium channels start to open. And then there is, we know calcium is more present outside, so it start to enter into the, after opening a few potassium channel, potassium leaks out and makes membrane negative, a little negative from inside. But meanwhile calcium channel, they get open and calcium start to enter into the cell and makes it stable at positive charges, because calcium are positive. So inside of the membrane was positive, it was depolarized, it goes a little toward negative due to opening of potassium channel, but at the same time calcium channel they have been open, and now calcium is entering into cell, making it a little more stay at positive charges. So this phase of the sustained positive charges on the cardiac muscles is called as plateau phase. So this plateau phase is not present in neuron and skeletal muscle. It is due to calcium ions and calcium channels that are only present in the heart muscle, not in skeletal muscle membrane and not on the neurons. They are not functional like that. And then there is opening of a lot of potassium channels, so that the entry of calcium is negated by the release of potassium from the cell. So a lot of potassium, because they were slow channel, so a lot of them get open, and then the potassium start to leak out and the membrane goes toward repolarization. And then at the certain level of negative ions, the calcium channel that were closed at resting membrane potential, they again get closed, so the membrane goes to the complete resting membrane potential. So there is difference of only plateau phase that is provided by calcium channels and calcium ions, entry of calcium ions into the cell. Heart is in special cases, in special conditions, controlled by sympathetic and parasympathetic nervous system. It is getting input from proprioceptors, from chemoreceptors, from baroreceptors in the blood vessel, chemoreceptors in the blood vessels, proprioceptors in the limbs and organs, and it is getting the information about blood pH. It is regulated by hormones like epinephrine, norepinephrine, and it is influenced by ions, age, gender, temperature. So these all things, they can change the beating capacity and the beating frequency and the force of beating of the heart. Uh, so in the we can see the involvement is only in special condition. If there is change in certain physiological conditions, then the heart muscles, they will change their beating frequency and the beating force. Otherwise they will be beating in a normal range for the uh, longer times without any input from the nervous system. Here we can see the heart center, or the cardiovascular center, is present in the medulla oblongata, and that is the region to control involuntary muscles, and particularly to the heart. So the input are coming to this center from the cerebral cortex, from the limbic system, that is the system of the brain for the emotions, and then the hypothalamus. And limbic system is for the basic instinct of the uh, life. For example, the emotions, the food, and the reproduction for the finding mate, etc. So for the thirst. So these information, they are coming from hypothalamus, limbic system, cortex onto the cardiovascular center in medulla oblongata. And it is also, information is coming from the periphery of the body, from proprioceptors that are monitoring the movements of the limbs. These are the for the positioning of the limbs and the organs, and chemoreceptor that monitor blood chemistry, baroreceptor that are monitoring blood pressure. They are present in the blood vessel that are going toward head, that are the carotid arteries. So these information, they are coming to the medulla oblongata. So medulla oblongata is well informed about the physiological conditions of the blood, and the uh, it is also informed about the positioning of the organs, and it is also informed by cerebral cortex, limbic system, and hypothalamus about the basic instinct or the for the survival, that is emotions, and the basic necessities that are food, sex, and the thirst. So then there are outputs. So motor neurons, they are going from the medulla oblongata of sympathetic nerves and parasympathetic nerves. So these sympathetic nerves, they are cardiac accelerator nerves, and then there are vagus nerves in the parasympathetic system. The parasympathetic system, it is to decrease the rate of spontaneous depolarization in SA node and decrease the heart rate. If heart rate is increasing, blood pressure is increasing, that input is coming from the baroreceptors, and if the conditions are normal, going to be normal, then they have to reduce the heartbeat. So it is the vagus nerve or the parasympathetic nerves, they are reducing heartbeat and the force of contraction of heart. And the if the conditions are getting worse, then there is need to increase rate of spontaneous depolarization in SA node and increase the heart rate, increase the frequency of nerve impulses. That is done by this cardiac accelerator or the sympathetic nerve. It is coming from medulla oblongata, and it is increasing heartbeat of the frequency of heartbeat and force of heartbeat of the heart, in order to, because it is getting information about the all these parameters. So in order to increase survival, the heartbeat is being increased. In otherwise, if these conditions are normal, then medulla oblongata will not send signal to heart to either decrease or increase heartbeat. It will be beating on a normal rate. So heart in normal condition is not requiring any input from the nervous system. But these normal condition, they are for very few times, because in the life of an organism in daily life, the emergency and conditions and normal conditions, they run side by side. For example, if you cross a road and you see a vehicle near you, then your heartbeat will increase. That is a special condition. In order to make your survival ensured, the heart has to pump more blood to your muscles of legs in order to cross the road, and to send more blood to your brain to make a decision, either you have to cross or come back. So uh, some other things, like if you take a lecture, and if you have to take a test, these all conditions may change heartbeat, and it is by the sympathetic or parasympathetic nervous system.